Good day everyone, especially to you Ma'am Dan. So today, we will tackle my assigned topic, which is all about gynogenesis, androgenesis, clone populations, and nuclear transplantation. So what is gynogenesis? Gynogenesis refers to the production of all female offspring by inactivating sperm. Gynogenesis is the process of embryonic development with solely the maternal genome and without paternal genetic input, a phenomenon similar to parthenogenesis. So here's the stages in oogenesis or the gynogenesis. Gynogenesis occurs in nature but can also be induced artificially. First, natural gynogenesis, and second is induced artificial gynogenesis. Natural gynogenesis is a type of asexual production where the egg develops into the embryo without being genetically influenced by sperm. However, sperm is required to activate the egg, but it does not contribute genetically to the offspring. As a result, the offspring are clones of the mother as they inherit only her genetic material. So, natural gynogenesis has been documented for some members of the family Pucelidae fishes, while the artificial or induced gynogenesis is a reproductive process where the development of an embryo is artificially initiated using a sperm to activate the egg, but the genetic material from the sperm is excluded or inactivated. This ensures that the resulting offspring inherit their entire genetic material solely from the female parent. Genesis has been successfully induced in several fishes. Notable among them are Ndan major carps, Labeo rahita, Catla catla, Cerus amargala, and Chinese carps. So next is androgenesis. So what is androgenesis? Androgenesis is a method for producing fish in which all of the nuclear genetic information originates from the male parent or from the sperm, while the mitochondrial DNA is still maternally derived. So how androgenesis works in fish? It works in egg inactivation, fertilization, chromosome doubling, and embryo development. What clone population does mean? Clone populations refers to groups or organisms, cells, or genetic material that are genetically identical derived from a single ancestor through the asexual reproduction or artificial cloning methods. So here's the examples of clone populations and natural clones. First is bacteria. Bacteria populations arise from single bacterial cell through binary fission. Second is plants. Plants form clones through runners, tubers, or root suckers. In artificial clones, they use the sheep or dolly. Cattle and even endangered species have been cloned using somatic cell nuclear transfer. So what is nuclear transplantation? Nuclear transplantation, which refers as somatic cell nuclear transfer. Nuclear of somatic cell donor transfer into an inoculated egg or oocyte recipient cell. Who can undergo the nuclear transplantation? We can undergo nuclear transplantation in animals. Do you know what is dolly? Dolly, the sheep, was the first mammal to be successful cloned from an adult somatic cell. Using a technique called somatic cell nuclear transfer, she was created by scientists at the Roslin Institute in Scotland, led by Ian Wilmot and Kate Campbell, and was born on July 5, 1996. Dolly was the name after the singer Dolly Parton. For human nuclear transplantation using somatic cell, there is no laboratory has succeeded. because of the safety and efficiency of the procedure.